Good morning, good afternoon, and good evening, everyone. My name is Janine Stevens. I'm one of the Janelia organizers of Life Science across the globe. And on behalf of all 8 sister institutes in this series, I welcome to today's event on cancer immunology hosted by the Tsinghua-Peking Center for Life Sciences in Beijing. But before we get started, I want to encourage you to visit lifescienceacrosstheglobe.org to see our full lineup of monthly events for the year, to subscribe to the calendar, and to also view recordings of past events. Also, I want to mention that we would be eternally grateful if you would please complete the brief survey that we'll post in the chat box at the end of today's session. And a reminder to trainees, please stay on the call after the event today for a special Meet the Panelists session with our speakers and our moderator. And audience, please use the Q&A box for your questions at any time during the event for the speakers, and we will ask them during the Q&A period. And now I would like to turn it over to Professor Zhang from Peking University for some opening remarks on behalf of the Center for Life Sciences. Zhang.
Hi, everyone. I will start with a brief introduction about the Center for Life Sciences, which was established as a joint center between Peking and Tsinghua University in 2011, with the goal of reforming education and research in China, attracting talented scientists across all career stages, and bringing together scientists from diverse disciplines, including biology, chemistry, physics, psychology, medicine, engineering, computer sciences, and so forth. So the mission of the center is to carry out innovative and cutting-edge research to address fundamental questions in life sciences using interdisciplinary approaches and to nurture the next generation of scientists and innovators. So there is a high-level cooperation between the 2 universities, which is very much encouraged for the faculties and also the students who can take classes from both universities. And our campuses are also next to each other, which makes our interactions very easy. So our funding directors were structural biologists, Yigong Shi, and neurobiologist Yi Rao, but they've now all been promoted to presidents of other universities. So last year we have our new directors who are biologists, Hongwei Wang and Chao Tang. They're our current directors. So the Center for Life Sciences is very pleased to be hosting today's Life Science Across the Globe event on cancer immunology. We have a very exciting group of panelists. So without further ado, I would like to introduce our moderator for today, Professor Padmanee Sharma from MD Anderson Cancer Center.
Thank you, and hello everyone. Welcome to this session. And our first speaker will be Dr. Aviv Regev, Executive Vice President from Genentech in the United States. So Aviv, take it away. Thank you.
Thanks, Pam. Let me share my slides. Thank you for the opportunity to be here today. They should be looking fine now. So I have to actually start with my disclosures. I'm a co-founder and I have equity in Celsius and Immunitas, and I was an SAB member in Thermo Fisher, Neogene, and Syros. And from August 1st, 2020, I'm an employee of Genentech. I'm going to talk today, like others, about tumors that you can think of as complex ecosystems that are made of different kinds of cells. And that means not just different cancer cells with different mutations, but also different types of immune, stroma cells, and others. And so the interplay between these different cells in the tumors can promote tumor growth, but it can also offer important opportunities for therapy. And really one of the most remarkable examples of this in the past decade has been immunotherapy. So one of the non-cancer cell types in tumors is CD8 cytotoxic T cells. These are cells that can recognize and kill cancer cells. But cancer cells can respond to this by using molecular mechanisms that we call checkpoint molecules in order to exhaust the T cells. And so a form of immunotherapy called checkpoint inhibitors inhibits this axis, and this unleashes the T cells to kill the cancer cells and bring benefit to the patients. Now, unfortunately, for some cancer cells and some tumors still find ways to evade this and to avoid T cell killing. And so as a result, only some patients benefit from immunotherapy. And in other cases, the tumors are resistant. One hypothesis has been that this resistance is associated with a difference in the T cell populations of the tumors. So some tumors might be cold. They don't have any infiltrating T cells, whereas the responsive tumors are hot and they're filled with T cells, which you see here in the color red. And so, in fact, scientists sometimes categorize now tumors by the portion of T cells and where they are located. At one end of the spectrum are these hot, inflamed tumors with a lot of T cells inside them. At the other end are immune deserts with no T cells inside. And in between are the immune excluded tumors where the T cells are at the periphery, but they haven't infiltrated the tumor. And so if we want to understand better cancer, but also treat patients better, we need to be able to look inside tumors and figure out the molecular mechanisms within and between the cells that drive it. And then, just like in the case of checkpoint inhibitors, we would want to intervene in these mechanisms and help the immune system do what it does best, which is to kill tumor cells. So ideally, we would be able to look at each individual cell in a tumor, cancer cell, immune, stroma cell, and so on, and characterize each of them by the molecules that they have, for example, the RNA that's expressed from each gene. But how might we go about it? Well, until quite recently, we used to do this by taking hundreds of thousands or millions or billions of cells in one tumor and measuring their average. And if you think of each cell in the tumor as a piece of fruit, this means we generated the lab equivalent of a fruit smoothie. In the last few years, a method that we developed called single-cell RNA-seq allows us to measure the expression of genes in each cell separately. So you can think about that as a fruit salad. And with more recent spatial genomics technologies, we can actually profile cells that are organized together in the tissue. So a fruit tart. Today, I'm going to focus mostly on single-cell RNA-seq. One of the challenges with methods like these is that they generate very complex, very noisy, and very big data. Each cell has about 20,000 genes in the genome. And if we characterize it as a point in a high-dimensional space where the number of dimensions equals the number of genes, it's not just 3 dimensions, it's 20,000. And that's very hard to reason about intuitively. But algorithms are really great at discovering the patterns in the data and reporting them back for different cell types and states and how they change over time and the gene programs that they use and even where they're physically located and how they interact with each other. So today I'm going to share with you several vignettes on how we use these kinds of approaches to try and dissect tumors and understand the relationship between cancer cells and the immune cells. And I'm going to start with melanoma, which is one of the first tumor types where there was a major benefit to patients from immunotherapy. So several years ago, Livnat Jerby and Ben Izar, when they were postdocs in the lab, used single-cell RNA-seq to profile tumors from 31 patients, half of them not yet treated and half of them after treatment. All of them were non-responsive. And Livnat and Ben wanted to know if cancer cells from some patients have properties that make the tumor better at excluding T cells and as a result more resistant to immunotherapy. So they pulled only the cancer malignant cells from their data. And each of these cells is shown here as a colored dot on these plots on the right. And it's positioned in 2 dimensions based on its profile. In the top plot, the cells are colored by patients. And because tumors are very different, each patient's cells form a separate group or cluster. But what we want to find is what might be shared between the tumors of different people. And so we're looking for these common features. For this, Livnat developed a computational method that looks across many tumors in order to find features in the profiles of cancer cells that correlate with the frequency of T cells in the same tumor. That means she's looking for genes that are expressed only in cancer cells, but they're higher, for example, when the T cell levels in the tumors are higher, or they might be higher when the T cell levels in the tumors are lower, so anti-correlated. The result was a coordinated gene program in the cancer cells, which we call a T cell exclusion program. And in this program, we have genes like MHC class 1 or interferon gamma that are low in cancer cells in cold tumors and high in hot tumors. And we have genes like MYC targets, CDK4, remember that gene, and CDK targets that are high in cancer cells in cold tumors and they're low in hot tumors. Then we wanted to make sure that we see this program in the original tumors. So we went back, to the fruit tart. We used a method that measures multiple proteins in sections from 19 of our patients. And this allows us to mark the key cell types and the exclusion program. So just to orient you, here's one section. In blue, you see the T cells, and the CD8 T cells are in light blue. Red cells are cancer cells that repress the exclusion program, so we expect them to promote a hot environment. And green cells are cancer cells that induce the exclusion program, so we expect them to be in a cold environment. And this is exactly what we see. The red malignant cells are interlaced with the blue T cells, but the green cells make these big cold patches. So now the bigger question was, would this program be related to the patient's response to immunotherapy? For this, we studied a new cohort of 112 patients in collaboration with Dirk Schadendorf and Eli Van Allen, where the patient tumors were biopsied before the patients were treated with checkpoint inhibitor and profiled by bulk RNA-seq. And then the patients were followed to see the clinical outcome. And we found that a higher expression of the exclusion program in these tumors before treatment actually predicted a worse outcome for the patients. So of course, we would want to find ways to inhibit this program and suppress it as a new therapeutic approach. So we used another computational method to analyze RNA profiles from hundreds of cell lines that were treated with drugs, looking for drugs that would be more toxic to tumor cell lines that express this program. And we found that CDK4 inhibition would repress this program. Now you might remember that I told you that CDK4 and targets of CDK4 and 6 were actually included in the exclusion program. So we tested this idea first in cell culture, we used cell line, and then we grew even malignant cells that we isolated from the tumors together with T cells from the same patient tumor. And treatment with a CDK4/6 inhibitor drug called abemaciclib reversed the expression of the program. And finally, we showed that treating animal models of cancer with a sequential combination of first checkpoint inhibitors and then the CDK4/6 inhibition reduced tumor growth. Now, at this point, we also asked ourselves, is this unique to melanoma, or could this be found in other tumor types as well? So we scored bulk RNA-seq data from multiple tumors in the Cancer Genome Atlas. And we found that cutaneous melanoma, lung adenocarcinoma, and kidney cancer all have relatively low scores. And that's consistent with the benefit of immunotherapy in these tumors. On the other hand, cancer types that are typically not responsive, like uveal melanoma and colon cancer, have relatively high scores. And the highest score was in synovial sarcoma, which we later showed also expresses this exclusion program. And it is also related there to actual T cell suppression and exclusion. And it turns out that in synovial sarcoma, the expression of this program is controlled by the main genetic driver, which is a fusion protein called SS18-SSX. So to summarize this first part, I showed you how we use single cell genomics to make a statistical prediction about a program in malignant cells that leads to T cell excluded tumors. We validated that it predicts the response to immunotherapy in 2 independent cohorts. And then we predicted that it can be reversed by CDK4/6 inhibitors. And we validated that in cell and in animal models. And then we also saw that this program is likely present in cancer cells of other tumor types, and we validated that in synovial sarcoma. But now, can we take a complex program like this and now try and take it apart and figure out what's going on? Now, in the first study I showed you, we really followed just one pair of genes, CDK4 and 6, out of dozens and dozens of genes in this program. And to be honest, this was because we conveniently had this drug that already inhibits them that somebody developed. But if we want to test every gene for its effect, we need to use a genetic screen where we knock out each gene in turn. And also we want to see the effect on a complex program of genes. And so we need to measure the impact on an expression profile. And then we need to conduct the experiment in the co-culture. We're going to genetically manipulate the patient's melanoma cancer cells, but we grow them together with T cells from the same patient in order to see the effect. And so Chris Frangi, Johannes Melms, Pratiksha Thakor, and Katie Geiger, in collaboration with Ben Izar, decided to pursue this using a method we developed a few years ago called Perturb-seq, where we grow all our cells together, perturb each of them genetically using a different genetic reagent, which is barcoded with a unique sequence. And then at the end of the experiment, we do single cell RNA-seq. And this gives us both the identity of the genetic perturbation and the impact on the profile of the cell. Now, we had one more remaining challenge, and this is because many of the best established ways to decipher what is happening to the cells is based on looking at proteins on the cell surface. And some of the most canonical proteins are actually very nicely detected on the cell surface, but their RNA is not sufficiently well detected by single cell RNA-seq. And so we modified the Perturb-seq method in order to rely also on a method called Cyst-seq, where we stain the perturbed cells by DNA barcoded antibodies, so that we can measure the level of these antibodies in the profiling as well. And with this new setup, we perturbed each of 248 genes in the exclusion program inside the patient-derived melanoma cells, and then we assessed the impact in two ways. One was using the cell viability in co-culture with T cells to see if T cells were killing the cancer cells, and the other was on the cancer cells RNA profile. And so first, in the cell killing screen, the strongest hits were in genes like beta-2 microglobulin, HLA, and the JAK-STAT pathway genes, which, when we perturbed them in the cancer cells, gave these cancer cells resistance to T cell killing. And this is what we would expect. So it's a nice positive control. But there were also additional genes that conferred resistance when they were perturbed, including CD47, CD58, and CDH19. And again, remember CD58. So now we wondered, are these working through the JAK-STAT pathway that we know would confer resistance, or is it a different mechanism? For this, we turn to the perturb-seq screens, where we analyze the profiles of 200,000 patient-derived melanoma cells and use the computational model that fits what we call a regulation matrix of the impact of each perturbation on the mean expression of each gene across the cells. And the most prominent portion of the regulation matrix, which I'm showing here, is consistent with the viability screen. JAK-STAT in the interferon gamma receptor inhibited a program of MHC and antigen presentation genes, chemokines and cytokines, interferon response genes, and so on. But their perturbation also induced genes like CD58 that I'll come back to. So after we had, again, a good positive control, we looked at the exclusion program genes. And when we perturb the JAK-STAT pathway genes, the program is increased. But perturbation of 22 other genes, including CDK6, MYC, ILF2, DNMT1, and ASCL3, repressed it. CDK6 is the one regulator that we already had before, but now we have multiple others that we could try and inhibit in order to repress this program. And so then we wanted to see how all the pieces wired together. For this, we looked at the rest of the regulation matrix of the impact of the perturbations on RNA and proteins. So on the other one hand, we can cluster the perturbations and we can find modules of perturbations that have a similar impact across the RNA and proteins. On the other hand, we can group the RNA and the protein profiles, and we can find programs of genes that are co-regulated across the perturbations. And altogether, this gives us 6 modules that impact 4 programs. For example, in module 1, you have the regulators SOX4, RB1, SP100, and IRF3 that activate a program of the cell cycle, metabolism, inflammation, and checkpoint genes. SOX4 and RB1 inactivation was already shown to impact the response to immunotherapy, and these regulators themselves are actually repressed when you perturb the JAK-STAT pathway, so it's downstream of that pathway. On the other hand, the same program is repressed by module 2 with genes like CD58 and CXCR4. Now, this is a very short talk, so I don't have time to go through every detail, but this model is like a map now for all the different mechanisms related to exclusion or resistance through this particular program that we started with. And to illustrate this, I'll highlight just one example, CD58, which I mentioned before and will now recap. So CD58 is part of the repressed side of the exclusion program in the patient tumors. CD58 knockout cells are more resistant to T-cell mediated killing in a viability screen. CD58 is not just activated by the JAK-STAT pathways, actually it's induced. The CD58 knockout has a different phenotype than the JAK-STAT knockout in the perturbed site-seq screen, and it didn't affect the expression of antigen presentation genes. And so altogether, this led us to hypothesize that CD58 loss is a distinct resistance mechanism. And CD58 is an adhesion protein. It's typically expressed on the surface of cells that present antigens, and it binds to CD2, another molecule, on CD8 T cells and on NK cells. And it has come up before in T cell killing screens, but there's little known about it, relatively speaking, because there's no ortholog in the mouse. And so we validated that CD58 knockout melanoma cells are resistant to both T and NK mediated killing. And we also validated the perturbed site-seq result that the CD58 knockout doesn't impact MHC class 1 or 2 expression, but it does induce one of these checkpoint mechanisms, PDL1, which may be one of the mechanisms by which it impacts resistance. And finally, we went back to the patient's original data, and we saw that it is downregulated in patient cells after immunotherapy. We've now gone global from 248 to the whole genome, with a genome-wide perturbation screen for regulators of MHC class 1 in melanoma cells, with another tweak to this perturb-seq method. So to summarize this second part, I showed you how we used perturb-site-seq to drill into the T-cell exclusion program. We parsed out this well-established JAK-STAT module, but also 4 other modules that govern 8 programs, including the exclusion program. And one highlight is the distinct role of CD58, but there's many other regulators and story in this map. Okay, so in the last couple of minutes, until now, we try to dissect the tumor to its component cells and then assemble them back together. And for example, we focused on cancer cells and CD8 T cells because honestly, we already knew that they interact with each other and that the interactions is important. But can we do the inverse? Can we use the data with no assumptions and look for multicellular units of cells that interact with each other and might be important for the tumor? And the short answer to this is yes, we can. And the basic idea is very simple. We expect cells to coordinate their action in the tumor, either because they're all responding to the same signal or because one of the cells impacts the others. And in any case, we expect this signal in the data to reflect the coordination. And to find this kind of signal, Livnat again developed a computational method, which we call dialogue. And here it learns a way of representing the cells in ways that show the dependencies between them, either across different physical niches or across samples or across conditions. So to illustrate this in cancer, here's data from a published melanoma study where 48 patients, 31 responders and 17 non-responders were profiled by single-cell RNA-seq, but this study profiled only the immune cells in the tumor. So we're going to focus on how they relate to each other. And here is one of the multicellular programs that we now discover, and it is associated with a response to immunotherapy. So the induced part of this program is higher in responders versus the non-responders, and the repressed part has the opposite pattern. But unlike before, where the program I showed you was made only of genes co-expressed in one cell type, here we have different genes in 4 different immune cell types that coordinate their expression. So as a result, the program connects macrophage M2 polarization, the repression of a gene called ApoE, and T-cell dysfunction. And it also includes a gene called CXCR6 as repressed in CD8 T cells. And we have in a different study shown that CXCR6 is a pan-cancer marker of dysfunctional CD8 T cells, and it's needed for interactions with myeloid cells in the tumors. I am pretty certain you're going to hear more about these kinds of cells and interactions from Miriam and Zemin today. And now it turns out that we can apply these approaches to many tumor types. So for example, here are 2 main types of colorectal cancer. 15% of patients have tumors with a lot of mutations, very hot with TILs, respond to immunotherapy, but 85% of patients have tumors with far fewer mutations, typically cold, and don't respond to immunotherapy. We went through the exact same thing I showed you. Lots of patients, lots of single cells, lots of programs. The difference is that we also looked at how the programs co-occur across the tumors for different cell types. And in doing so, we found 6 major multicellular communities, including an inflammatory luminal hub of specific cancer, immune, and stroma programs in both tumor types. But we also found a unique anti-tumor immunity hub, which is only present in these inflamed tumors that respond to immunotherapy. And there's a lot of upcoming methods that are going to help us characterize these kinds of communities and their mutations. So to summarize the final part, I showed you how we moved from cellular to multicellular programs that relate different cell types and their programs across patients, niches, or conditions. Some of these multicellular communities, like some of those in melanoma, colorectal cancer, there's also pancreatic cancer and more, relate to the patient response to immunotherapy or to survival. And upcoming experimental and computational methods will help us better discover these multicellular programs and understand their role in tumor immunology. So I told you today about how profiling single cells helps us understand immune phenotypes in tumors, dissect their cellular component, dissect their molecular circuits inside the cells, build back the multicellular communities, and help us ideally predict patient outcomes and identify new targets for therapeutic intervention. I want to thank all the people who did the work. I mentioned them throughout, and I look forward to questions and the discussion later on. Thank you very much.
Thank you so much, Aviv. That was super exciting, and we're looking forward to the discussion in the panel. Now it's my pleasure to have Dr. Miriam Merad, Director of the Precision Immunology Institute at the Icahn School of Medicine at Mount Sinai, to give the next talk.
All right, so it's a pleasure to be here today, and I'll be talking about the big effort in my lab to identify novel immune target of cancer, focusing on a specific immune lineage that I'm going to try to introduce in the first part of my talk. So Aviv introduced that immune cells are really a part of the tumor microenvironment, and this is what I'm highlighting here, is that all these different immune cells are really sometimes dominate the tumor lesions. So immune cells form a large network of cells with distant and synergistic functions, and there is 4 main tasks that the immune system is trying to accomplish. And this starts with the recognition of a threat. So it has to recognize a threat, and the threat could be the tumor cells or in infected cells, respond to a threat by producing inflammatory molecule, recruiting effector cells, remember a threat, and this is what we call this memory response. That's quite useful when we want to react faster when the threat occurs again, but also regulate a response, because the risk is always to have an overblown immune response that can lead to potential autoimmune response. And this is what the immune system is always trying to do, and this is why the immune cells are present, in fact, in most disease lesions, from cancer to atherosclerosis to neurodegenerative disease. There is always an inflammatory component that really is present in disease lesions. And I argue that targeting inflammation should be part of all treatment against all human diseases, because there is always, this inflammation is always contributing to disease outcome. Okay, now T cells are an important effector arm of the immune system that have a dedicated machinery to eliminate damaged cells, in this case, the tumor cells, in a very specific manner. So here, for example, you won't be able, I think, to see my little video. That's a shame, but what I wanted to show you, maybe I can do it this way, is that the T cells here in gray have been educated to recognize cells expressing a green fluorescence protein. This is the tumor in green here, and they are going to kill the tumor cell that express the GFP, but there is another tumor cells here that lack this green fluorescence protein, and well, the T cells cannot kill it, right? And this specificity of killing is what cancer immunologists are trying to really harness, right? However, what's important to realize is that T cells cannot recognize tumor cells on their own. They have to be educated to recognize a threat. And there is 2 cells that really are going to educate T cells. And these are dendritic cells and macrophage, and we call them antigen-presenting cells. And what they do is that they capture damaged cells, in this case, the tumor cells, process it in a way that will be recognizable by T cells. So this is a very important basic immunology principle that's important to really understand when we think about T cell modulation strategy. The cells that are able really to educate the T cells, not only to recognize, but also the type of effector program that T cells are going to acquire, are these 2 cellular compartments, dendritic cells and macrophage. And my lab has been working for more than 3 decades now, 2 decades in my lab, me for 3 decades. I'm trying to really understand the biology of these cells and try to really harness their biology to modulate an immune response against threat, both cancer and inflammatory disease. Now, while they can instruct, these same cells also regulate a response. This is a major task of the immune system that has been understudied in immunology. We study a lot activation, instruction of infector program, but we have not studied enough this regulatory program. And these same immune cells, right, while they are instructing T cells to act, they also make sure that T cells don't act too much by really inducing and engaging checkpoint molecule, which limit T-cell activity. And Jim and Allison and Honjo have shown that by inhibiting this checkpoint molecule, you can rescue T-cell effector function, and you can, in fact, rescue also T-cell ability to kill a tumor. But we know also that there are many more molecules that can do that beyond the PD-1 and CTLA-4, which are home run, so we may not be able to find another home run, but there are many other molecules that are regulating T cells. And I will argue that these APCs are the ones that really are going to shape or regulate much more potently your T cell function. So current clinical checkpoint blockade strategy target these two checkpoint blockade. There is more than thousands of immune molecules that regulate effector immune cells. So there is enormous opportunity to identify novel immune target of cancer, novel combination therapy, and also biomarker of response to stratify patients and maximize response to treatment. And I argue that targeting this antigen-presenting cell compartment is key to this endeavor, and this is the focus of my lab. So we focus on these two lineages of antigen-presenting cells, macrophage and dendritic cells. Both of them share this ability to capture, process, and present tumor antigen or antigen from infected cells to T cells, but they also differ. There are a lot of differences and subtleties, some that are subtle and some that are less subtle in their ability to shape an immune response. So dendritic cells are unique in their ability to instruct a T cell program. They can instruct from scratch. They can take a naive T cells and really do whatever they want with them. Macrophages are less potent to do that, but they are very potent at shaping the microenvironment because they, in addition to their ability to interact with T cells, they also produce many molecules that are going to shape the immune microenvironment by recruiting different types of immune cells, but they also produce metalloproteases and metabolites and contribute to the development of new neovessels. So they are a cell that really is a tissue cell that have in addition this immune function, whereas dendritic cells, which I always argue have developed more later during the evolutions, are really a sharp focus on modulating T cell function. And today I'm going to talk mainly about these cells. Although now I realize that for Ron, I should have talked about macrophage. That's okay. All right, so dendritic cells. So while we have learned a lot about the T cell molecular features that are conducive to effector anti-tumor immunity, much less is known about these macrophage and dendritic cells. And really to fill this gap of knowledge, we have built in my institution what we call a neoadjuvant research group. And this group, which consists of surgeons, pathologists, interventional radiologists, and cancer immunologists, are thinking about really human cancer lesions that can be most informative in our quest in understanding the molecular immune features that are conducive to anti-tumor immunity. So what we decided to do is spend a lot of time really profiling treatment-naive surgical lesions because this enables us to reduce confounding variables that are induced by prior therapy. We are working on an intact immune system because the patients have not been bombarded by steroid and chemotherapy. And this is how we are going to build our knowledge base. And then we take the surgical cancer lesions and we try to profile them using the technology that Aviv introduced so elegantly earlier, right? So we are going to really try to look at all these cellular programs that are present in the treatment-naive lesions. Okay, so this is, so we build quite an effort in profiling 3 main tumor lesions, lung, liver, and colorectal cancer lesions using different type of single cell technology, including CytSeq. And I'm not going to go into in detail how we do that, but what we focus on is the immune compartment mainly that is present in the cancer versus the adjacent tissue, which is tumor-free. And we are going to try to identify molecular programs that are enriching the tumor lesions versus the adjacent tissue. And here, this is a high-level view of this molecular program that are annotated by immunologists. So what we do when we look at this molecular map, if we spend a lot of time using prior immunology knowledge to annotate this cluster, and I will argue this is a very important effort, right? You should not just subcontract your analysis to computational biologists, but this should be really an integrated effort by expert immunologists that have been working on a specific pathway for years and computational biologists. And this is this type of integrated hub that we have built in our, what we call the target program. And so one thing that we've done, and this is a side discussion I wanted to have, is that we compare the program, the molecular program between the tumor and adjacent tissue versus the molecular program across different tumor. And what we found is that there is much more similarity in terms of molecular immune program across tumor than between the tumor and adjacent tissue. And this is I find a very hopeful finding, because it suggests that somehow first, it's not so surprising because checkpoint blockade is working across different tumor types, but it suggests that in fact, there is a commonality of response across tumor that we could harness. And what also is interesting is that we find that now when we go and look at the molecular profile performed by different groups, we can see that because of this unbiased profiling, now science suddenly is much more reproducible, right? So we compare the cluster the immune program that we identify in our Mount Sinai cohorts. Mount Sinai is close to Harlem. We have many very strong ethnical diversity compared to other molecular profiling performed, for example, by this Belgium group. And we can see that we can now find very similar program across these different cohorts. Here it was a lung cancer lesions. So again, this suggests that this commonality of response that we could harness. All right, so now I'm going to dive a little bit on dendritic cells. So I'm going to do hardcore immunology, and if it's too difficult, then we can maybe talk about it in the discussion. So now we are going to look and dive in these two cellular compartments, macrophage and dendritic cells. And we look at different programs that are accumulating in the tumor versus the adjacent tissue. And I've been working on these cells for a long time. So, we know how to recognize, something that is really out of the ordinary. So we start to annotate these clusters, and we find, for example, these different population of dendritic cells, and in fact, some of them was identified by my laboratory. And we were intrigued by this cluster of dendritic cells here. Here the effort is to find molecular programs that are dampening or limiting or restricting APC function, right? That can give us a hint on DC ability to really reduce or limit anti-tumor function, right? So we are looking at programs that can help us, and understand, their role in tumor lesions. And we are very intrigued by this dendritic cell cluster that we found by this single cell profiling strategy. So we find it in lung cancer. We also found it in liver cancer. We also find it in colorectal cancer. And now we had found it in all cancer that we've looked at. So it was a very interesting cluster. And so the first things we did is ask whether the same. So now it's difficult. So what do you do when you profile? And one thing that you can do is go and try to understand the function of these cells ex vivo, or you bring it to a mouse model. And this is what we did here. We go and do the same exercise of profiling in mouse lung cancer lesions and ask whether this same DC cluster is present. And indeed, we identify the same DC molecular program in this mouse lung cancer lesions. So now I'm going to just describe what this cluster express. And what we find is that this cluster of dendritic cell express many maturation molecules. These are molecules that are very good at activating T cell, but also express a very strong regulatory molecule. And what was striking is that this DC expressed very high level of this checkpoint molecule called PDL1, which engage PD1 on T cells, but also many other regulatory molecules. And this is what we were excited about, because potentially we could harness to modulate T cell function. This DC also express migration molecules. So this is also something that's very interesting to us because dendritic cells, when they are activated, they migrate where T cells are, which is lymphoid organs, either lymph node, tumor draining lymph node or lymphoid structure in the tumor lesions. So we call these cells MREG for maturation and regulatory molecule. And then we spent a lot of time trying to understand how this program was induced. And what we found is that this regulatory and maturation molecule was induced when dendritic cells captured tumor antigen. And this is what I'm showing you here. I'm saying that I'm out of time, so I have to go a bit faster now. So this program was induced when dendritic cell ate tumor antigen. So now if they ate, usually they have to present it and present it to T cells. So the next question we ask is whether this program was in fact interacting with T cell. And we use two strategies. We use this PIC-seq strategy that was developed by Ido Hamid's group. So one of the postdocs of Ido Hamid from the Weizmann spent a few months with us at Sinai. And then using this technology that purified, in fact, doublet cells. You take a human tumor lesions and then you purify cells that express both myeloid marker and T cell marker, and then bring it to the single cell sequencer. What we found is that in this doublet, T cells that were, the DC that were interacting with T cells were in this MREG program. So the MREG are the DC that are interacting with T cells. I'm going fast here, I'm sorry. And then we went and used spatial transcriptomics to ask where this MREG program was present. And here, with the help of our pathologists, we first annotate these tumor lesions, and then we are going to project this different program. And what we found is that this MREG program was specifically enriched in tertiary lymphoid structure. Well, we are very interested in this structure because these are lymph node-like structure that we know are recruiting T cells to the tumor site, and these are abundant with T cells. So if MREG are here, it means that they are playing a key role in shaping T cell function. Okay, so what I've showed you, I'm going to go fast here. What I've showed you is that this MREG program is induced when dendritic cells eat tumor antigen. They are really interacting with antigen-specific T cells, and we believe that if we focus on this program, we can identify relevant pathway for the modulation of antigen-specific tumor immunity. Maybe I'll have to stop in on this slide. So now, the beauty of single-cell profiling is that it helps us resolve really the molecular composition within cellular compartment. Now we realize that the same dendritic cells has at the same time both regulatory program and immunogenic program, and it's all about this balance, right? And this is really the definition of an immune response. An immune response is always the end result of a balance between activation and regulation. So now what we are doing is trying to really understand this balance better and try to really target programs that can help us shift toward immunogenicity, right? And couple this tolerogenic from the regulatory program within the same cellular compartment. And we are, and an example of that is working on this balance of interferon gamma receptor and IL-4 receptor. What we found is that these dendritic cells express very high level of IL-4 receptor. Now, IL-4 is a type 2 cytokine that is associated with allergic response and usually dampen what we call type 1 immunity. That's very important to get rid of tumor cells and virally infected cells. And what we did, so I can stop here. And what I was going to show you is when we block interferon gamma receptor, we dampen this immunogenicity. When we block IL-4, we enhance tumor immunity. And this is very, very interesting to us because we realize that this type 2 immunity, maybe I'll show you something here, blocking IL-4. But we realize that this IL-4, which was thought to be an allergic response, is in fact present in many epithelia. It's a primitive response not to induce allergy, but really to respond to different type of injury. And it becoming really a dominant program in my group now is really understanding type 2 immunity in the context of anti-tumor immune function. Okay, so I'm going to stop here, unfortunately, because, right? Do I have to stop, Pam? I have to stop, no? Or should I just?
It would be best so that we can move on to the next talk and have time for
Okay, I'm going to stop here. All right, so I just wanted to just say that the big focus is really this immune balance, maybe just like show this composition, understand this immune balance, the balance of molecular composition, really trying to shift. And now we have a big focus on really trying to dampen these type 2 response that are dominating many tumor epithelial compartments. All right.
Thank you so much, Miriam. I'm sure we're going to have lots of exciting discussion in the moderated panel. Now it's my pleasure to introduce Dr. Zemin Zhang, professor at Peking University, to give the next talk. Thank you, Dr. Zhang.
Thank you very much. And it's a great honor to have the opportunity to present our work in this very interesting online meeting, especially this presentation was done together with Aviv and Miriam, Merad, the two scientists I respect the most. So what I plan to talk about is the tumor microenvironment and how they are related to immunotherapies and how different cancer types may have different properties in the tumor microenvironment. Before I do that, so let me just show my disclosure. I'm essentially involved in two different companies, founder, scientific advisor, Analytical Biosciences, and I'm also a board member for InnoCare. So as we have known from the previous two speakers that the tumor microenvironment is a very complex tissue. Inside the tumor, it's not just a collection of tumor cells. In fact, it's very complex. There are many immune infiltrations in the tumor. And so we actually need to understand the tumor microenvironment in a very detailed fashion for us to define strategies to perturb the tumor microenvironment to allow immunotherapies. So in the past few years, my lab has been focusing on answering some basic questions about the TME regarding their specific immune compositions, if you have any special functions, where do the cells come from, where do they go in the end, what are the cell-cell interactions like, and most importantly, how they are related to clinical parameters, including how they respond to immunotherapies. So here essentially is the outline and the key points in my talk. Number one is that I want to mention that TME harbors multiple unique properties, which is also illustrated by the two speakers. And we can find multiple cancer-specific cell types. And also different cancer types may have very distinct properties in the TME. And I also want to mention our work on understanding the various factors influencing TME. And finally, I want to describe how the cell types may be related to immunotherapy responses. So here is probably not a surprise now that single cell is extremely powerful, as illustrated by Aviv talk. So essentially what we are doing is take of a given tumor types, we will take multiple cancer-related tissues ranging from tumor-adjacent normal tissues, lymph nodes, ascites, blood, and so on and so forth, to profile, go through the single-cell sequencing so that we can understand the unique cell types, the tissue compositions, cell-cell interaction, the marker gene identification. Then you can repeat this for multiple tumor indications so that we can have a more systematic view of the tumor-related tissues. So by doing this, we essentially started by studying the T cell properties. Then we gradually broadened to include all tumor immune types. So what we found early on is that different cancer types, ranging from colon cancer, liver cancer, and lung cancer, if you simply look at the blood samples, the differences between them is actually very small. But if you look at the normal tissue, then the T cell composition are actually very different, indicating that tissue themselves play a very important role shaping the infiltrating immune cells. But if you look at tumor themselves, on top of the tumor-related differences, there are more dramatic differences. You start to see many tumor-specific cell types. So it seems like the tumor microenvironment may be influenced by the combination of the tissue location as well as cancer cells. From those, you can identify different immune cell types that seem to be specific or at least enriched in tumor. For example, we talk about, we all know the exhausted T cells are heavily enriched in tumors. But in addition to that, we also see a lot of activated Tregs in the tumor, and also the Th1-like cells, and the LAMP3DC, which I believe is what Miriam talked about as MREG-DC. In addition, you have SPP1 plus tumor-associated macrophage here, or C1QC10 here. So all those cells have very preferential presence or very specific in tumor location. These actually provide great opportunities to find novel targets, because if those are specific enriching tumor, that really means they're doing something special in tumor. Either they are provide a more of a pro-tumor function or a pro-inflammation kind of function. So these provide opportunities for us to find specific genes in those cell types to find ways to perturb those cells. So indeed, for example, if you have exhausted T cells, this implies, of course, you can try to reverse or prevent T cell exhaustion. For activated Tregs, which provide the more suppressive role in the tumor microenvironment, we will try to deplete those activated Tregs, or maybe we can try to activate those more beneficial cell types like Th1-like cells, or maybe modulate such LAMP3DC or MREG-DC here. Okay, so some of the strategies targeting these cell types have been very successful in the clinics. Some others are in the trials. So we want to take it one step further and ask a question that would all cancer types benefit in the same fashion by some of the strategies. So we have found that some of the cell types, when they are found, they are typically found in a one given cell cancer indication. For example, we initially identified the LAMP3DC from liver cancer only. For the tumor-associated macrophage, we identify SPP1 macrophage from colon cancer. And so those are found in individual tumor types. We want to ask a question, if those are universal or specific to some of the cancer types. How do you answer that question? The way to do that is actually to study multiple cancer types at the same time. That's why we started several types of pan-cancer analysis of infiltrating immune cells. For infiltrating myeloid cells, for example, if you simply profile multiple cell types, you can find that some of the cell types, LAMP3DC, seem to be universal in all cancer types. Some other cell types, like SPP1 macrophage here, are only detected in about 50% of the cancer indication. So these seems to be more cancer specific. But even for some of the cells that appear to be universal, their abundance may vary a lot. Again, LAMP3DC, if you look at their distributions across all these cancer indications, there is dramatic differences. NPC has a lot, stomach cancer has a lot, but other cancer cell types don't have a lot. So if you don't have a strategy perturbing LAMP3DC, then these cancer indications or patient selection should be factored in here. Similarly, we can look at the T cells in a more broader view. So here we essentially constructed a pan-cancer T cell atlas by collecting published T cell data set and also generating our own data sets. We constructed a very broad pan-cancer T cell atlas covering 21 cancer types from over 300 patients here. Now, we take those CD8 and CD4 cells, we can divide those in multiple different clusters. Again, you can see that terminal exhausted T cells seem to be very specific in tumor, and activated Tregs also seem to have very strong cancer preference. So here, just to give some examples here, exhausted T cells is actually one cell types that we pay a lot of attention to, but their presence are again, not quite universal. Those cell types seem to be highly abundant in liver cancer, esophageal carcinoma, head and neck cancer, renal cancer, but not so much in multiple myeloma and breast cancer. So again, this may impact how the therapeutic strategies should be chosen for different kind of cancer types. Activated Tregs again, very important, but those are more abundant in nasopharyngeal carcinoma, liver cancer, and so on and so forth, but not so much in breast cancer and stomach cancer. Okay, so some other cell types seem to be more immune specific or cancer type specific. So this should be kept in mind when developing more precise immunotherapy strategies. But we also ask the question, what really causes such differences? And what makes these cell types more abundant in one location versus another? So we start to understand this. It actually turned out to be not an easy question to answer. So with increasing amount of data spanning through multiple patients, we can start to draw the kind of correlations of different factors that may be related to the abundance of different cell types. So here, using a pan-cancer T-cell analysis, we start to see, what are the other indications, parameters that may be correlated with the presence of the cell types. But we found a small number of correlation, not many. For example, here, we found out that the BMI index seems to be correlated with the frequency of TFH cell types. And the tumor mutation burden, of course, is a very important factor that's commonly used as a biomarker for immunotherapy responses. When we look at how is the TMB related to other cell types, the only thing that seems to be correlated is the Th1-like cells. So this might be important because we all know, why would the TMB be important for immunotherapy efficacy? How is that related to T cells? We think that maybe Th1-like cells maybe serve as a link between the TMB and the T cell properties. But still, you can see for most of the cell types, we still don't really know how their frequencies are related or determined by different factors. So we need to find a way to really understand this, but how do you do that? It's hard to compare the TME properties across tumor. For example, if you take the tumor samples on colon cancer patients and liver cancer patients, it's hard to compare those head on because there are so many differences. You're dealing with different cancer types, you're dealing with different tumor niche, and then also dealing with different genetic background because those are from different patients. But perhaps we can come up with scenarios where we can somewhat reduce the level of complexity so that we can directly compare those. The colon cancer with the liver metastasis is actually provides such an interesting model because you have largely the same cancer cells in two different locations, but then also you have the same genetic background, but two different kind of tissue niche. So if we take those tumor samples and survey multiple different immune-related locations and by single-cell sequencing, then perhaps we can have the data. We can compare such data with patients with only primary tumor in liver or colon locations. Okay, so here we have the single-cell data. Then we work out a bioinformatic tool and develop a tool called PhenoAligner, which essentially take a cell from metastasis sites. When we compare which of the cells with cells derived from other locations, then we essentially use a k nearest the mean, k nearest the neighborhood methods to identify what are the other locations that provide essentially the same a similar type of transcription patterns. In this way, we can match up or align the phenotypes in one location to another in order to identify if the cells from the metastasis side is more influenced by the each of the by cancer cell types or the tissue location. So by doing this, we actually found two different patterns that came from the cross comparison. We defined some of the types as M type. Those seem to be associated with malignancy or cancer cells. For example, you have exhausted T cells, the Th1-like cells, and also activated Tregs, SPP1-TAMs. Those cells seem to be more influenced by the cancer cells themselves. Then we have other immune cell types that we define as N type. Those are associated with niche or tissue location. Those cancer types or cell types include cells like mate cells, the intraepithelial leukocytes, the NK cells, and some subsets are the macrophage cells here. Okay, so you can see that cells can be differentially impacted by various internal and external factors here. But those are derived from the cross comparison from tumor samples from the primary liver cancer, primary colon cancer, and the colon cancer with the liver metastasis. Okay, so you can see that the factors that influence in the tumor microenvironment may include cancer cells themselves. These may include specific mutations or tumor mutation burden, or from the tissue niche. That these include some of the tissue-specific factors, or some other genetic factors, or SNPs, or BMI, or maybe age and sex. But perhaps the factor that may be more interesting is how they be impacted by different therapeutics. So, the final few minutes, I want to talk about our work on studying the dynamic changes of tumor microenvironment during immunotherapies, with the goal to identify the TME factors that seem to be associated with responses or resistance to therapies. And also we want to identify how those the TME remodel itself during the therapy. So how do you do this? We want to study the dynamic changes of the TME during the therapy. So here's one example. If you take lung cancer patients treated with pembrolizumab or the anti-PD-1 antibody, you can take a biopsy sample before treatment and after treatment, and you perform a single cell analysis, and you try to figure out what are the individual cell types that seem to be correlated with responses, and what are the cell types that seem to go through the major changes during the therapy. So I'm not going to summarize some of the complex study into very simple plot here. So for example, if you take what we define as the exhausted T cell precursor, and we'll very often call those CXCL13 positive CD8 cells. Before treatment, these have a very low level, but after treatment, the responders would have much increased level of such cells, but not so much in the non-responders. So this actually can serve as a very effective predictor for drug responses. So how, in a way, so essentially the T cells would go through the T cell precursor before they change to exhausted T cells. In a way, it seems like when the patient receives the therapy, you're essentially preventing such cells from turning into the terminal exhausted T cells. So we think that, what is the reason, where do these exhausted T cells come from? We think that maybe there are some kind of pre-existing but small amount of text precursors that are either inside the tumor or from the blood. Upon treatment, such cells that go through proliferation, they actually stop going to the exhausted T cells. So we call this process the clonal revival process. Then you say, wait, what we've learned is clonal replacement based on the excellent work by Howard Chan's group. We know that at least for some cancer indications, the effective therapy is more related to clonal replacement. But we think that the both scenario can work in the cases where the patient would have a smaller, lower level of the tumor reactive T cells. What you observe are primarily the clonal replacement phenomena. But the patient who already have a large number of baseline tumor reactive T cells, like lung cancer, in fact, we do see a lot more a clonal revival observation. And such pattern can also be seen in other cancer types like triple negative breast cancer. Again, here we use breast cancer patients treated with either paclitaxel or atezolizumab, the anti-PD-L1 antibody from Genentech, plus paclitaxel. Then again, we take the needle biopsy before and after treatment. Then we study the dynamic changes here. So in a way, you generate single cell data. You get the single cell data before and after the treatment, and then we can try to identify the kind of cell types that at the baseline seem to be correlated with the tumor shrinkage. The second parameter we use is to look at the changes of such cells during the therapy to see what are the cells that seem to respond the most during the therapy. So what are the cell types that seem to be related to therapeutic responses? So now if I simply look at the in two different scenarios. We have responders and non-responders. We have pre-treatment and after treatment. And if you simply look at the immunotherapies, and between responders and non-responders, seems like the responder would tend to have a large level of baseline CXCL13 positive CD4 and CD8 cells here. And in non-responders, they don't have so much. And after treatment, the responders maintain higher level. It's actually proportionally higher level of such cells during the therapy. So it seems like the CXCL13 positive cells both predicted and mediated the effective responses to atezolizumab blockade. Okay, but interestingly, for chemotherapy, so it seems like even for responders, such cells actually depleted. So these cell types are actually proliferating, and the chemotherapy would selectively kill the cells. So it seems like what we think is what is going on is that these kind of cells are probably what's needed for immunotherapy like anti-PD-L1 to work. But unfortunately, if you have chemo added to the process, they actually selectively deplete such cells to make the combination therapy actually worse. So that's a kind of theory. And we also identify other cell types like B cells that seem to be related. And B cells have been linked to immunotherapy responses by multiple different groups. What we have found is that it's not all B cells that seem to be relevant. It's actually there is a particular B cell subtype called follicular B cells that seem to be exhibit a very high predictive index here. But we think the importance of the B cell is not necessarily their ability to make antibodies, it's actually their ability to regulate and activate other T cells. So we think that what's going on in the triple negative breast cancer situation is that, when you have the chemotherapy alone, the chemo would actually selectively kill the proliferating cancer cells, but along the way, they can also remove some of the beneficial T cells. And in some other, when you have the anti-PD-L1 antibody here, if you don't have a lot of CXF13 positive T cells, you probably don't have responses. And in a way, when you add the antibody, actually you keep such a T cell at a higher level. In a way that we think that those kind of T cells are actually the very important cell types mediating the effective therapies. So just to summarize, and we talked about the complex tissue microenvironment, and we talked about multiple tumor-specific subtypes that may provide future opportunities for find novel targets. Because once you find tumor-specific cell types, then you can find ways to maybe to perturb such cells, either inhibit or eliminate or activate such cell types. And once you develop the therapeutic strategies targeting cell types, then you can factor in the cross-cancer differences, because different cancer types harbor distinct immune properties. In that case, we need to have refined clinical strategies. Okay. So lastly, I just want to acknowledge multiple people who are involved in the work I presented here. I'm not going to name one by one, but here we have our website. It has more detailed contribution from different individuals and our collaborators. Thank you so much.
I think now it'll be great if we can just have a session where we have free dialogue, and I have a few questions that I'd like to pose to the panel, and maybe I'll start with you, Aviv, given the fact that, we've now heard these multiple talks about the plasticity and regulation of the immune response. How can we hope to integrate the power that you showed us with the single cell RNA sequencing and other approaches, of course, the computational approaches, but try to also capture the biology of this dynamic process so that even when we're looking at specific genes that may say, okay, this gene is the hallmark gene for a repressive program, but maybe that gene is actually not being expressed because the protein expression may be very different as the cell is going through its plasticity and its different states. How do we try to integrate the different technology advances to get better at capturing that dynamic process of the immune system?
So that's a fantastic question. I'll start with the fact that one of the strengths that the computational technologies give us is that we don't actually have to put all our hopes in one or another gene when we try to understand what's going on. but we can work at this level of multiple genes together, a program, or as I showed, even multicellular programs across multiple cell types. The result of that is that we get actually a signal that is more robust to the, not just to the differences in the biological levels of regulation, RNA versus proteins, but also just in the capabilities of the experimental assays to give us the most precise information. So by looking at the level of multiple genes and big profiles, we actually get more robust answers. This, of course, then switches when we have to start thinking about, say, therapeutic development, because there we actually do need to choose specific things that we want to go after. And I think it plays out in two different ways. When we go after a molecular therapeutic, you need to find the mechanism. It's not enough to just say there are cells of this kind. You need to figure out what is actually going on in these cells. And that's where we need to triangulate from three different perspectives. One is the patient samples that really figured prominently in Zemin's and Miriam's and my talk, because we have to start with the human biology and the human as the model. Then the ability to do experiments in a dish with patient-derived models like cells co-cultures, organoids for some tumors. That's very advancing very rapidly. And finally, of course, use the animal models as well. And by moving between the three of them, we can add interventions and perturbations. It can be drugs that we treat with, but it can also be genetic manipulation. And that allows us to understand the molecular causes. So that's, I think, the second perspective. And then the third one that comes back is when we try to do therapeutic development with those targeted therapeutics that we develop, we come back to the same approaches that we started with, because now we can look at the human patients in a much higher and more sophisticated resolution that we were afforded before. Before, you did have to choose a single biomarker. And if that choice had any limitations, technical limitations, the signal was not as great, it kind of worked, but it wasn't the perfect representative for all aspects. That was it. Now we can work with much richer things as our biomarkers, and that helps us understand better what is happening in the patients when we treat them and stratify them in more sophisticated ways and so on. So I do think it plays out in different ways at different points along the process of understanding what is going on, but it puts us in a different place than we were before. And the technologies keep getting better. Single cells, that's like so 2011. Now most of us are using spatial analysis approaches so we can see how they relate to each other. Just doing RNA, again, with clinical samples, you had to collect them 5 years ago in order to have answers now. So that's what people see today. But if you ask us about the experiments we do today, it's RNA and protein and the organization of the chromatin, multiple things all being measured together. So the technologies advance extremely rapidly right now. And that means that we would be able to know a lot more quite soon.
So I think this brings us to the point that you said about humans, right? You have to start with the model system that's going to be the most meaningful, and getting those samples becomes so important, and getting them from all of the different places that we talked about, right? They're not just a primary tumor, but the metastatic lesion. Then is the metastatic lesion in bone? Is it in brain? Is it in liver? Because these are all very different niches. So Miriam, maybe you can talk to us a little bit about how do we make that happen? Because you've put together this great team, that we saw in your slide, but how do we make that the forefront of everything we're doing? There are over 2,000, maybe even 3,000 clinical trials right now that are ongoing with immune checkpoint therapy agents. How have we really brought this to the forefront of the pharmaceutical companies and the academic organizations that reading out the clinical endpoints without putting together these teams that can give us access to these samples? We're doing ourselves a disservice that way. How do we make this better?
Yeah, so it's a very good point, and now Aviv, could you potentially even comment on that, because she's has a cross, to the other part of the aisle.
I'm going to get back to her.
Exactly, but I will even say, something we say now is that it's almost unethical not to do ancillary study or biological studies on these clinical trials, because as we know, and Aviv will comment on that much better than me, is that most phase 3 trials fail, and unfortunately, there's a lot of failure in these phase 3 trials. In fact, the way we called our group was a group that will help, the way we define our group that is engaged into this immune profiling of clinical trials. And now we are doing it, we are using the same type of group to go and profile patients during treatment, is that this will help us de-risk the phase 3 trials. It's really, in fact, we have a little commentary that's going to come up in Nature Medicine this week, where we describe that, not only in cancer, Pam, right? Across medicine, because in cancer, we are much, I think the industry is much more advanced. Like they are absolutely, they agree, they're so well, they realize there's so many molecules, they need a little bit of help from biologists to really make things make sense of all these different type of immune molecule. But in other disease, so I'm trying to do the same in inflammatory disease, and there is much more resistance, right? So you will do all these very expensive clinical trials. You are going to bring a new molecule to a disease. You have the potential to use this brilliant technology that Aviv has discussed, which will cost probably a fraction of the cost of the clinical trial. And yet, there is resistance from industry. So I think industry has to step up, and they are in cancer, less so in other disease. The other things is that our group, there are other resistances, sometimes the surgeons or the pathologists. The pathologists are extremely protective of their tissue. And when they protect to store the tissue, not so much to really do a lot of research on the tissue. So I understand there is legal procedure you have to bring and you have to store the tissue, but there is so much you can do with this tissue. So we have to change the mindset of our pathologists. They have to realize that they need to share. They need to share with scientists that, of course, are going to make a good use of this tissue. And then the third thing is we need to think of what are the relevant tissue pipelines, because sometimes the tissue is not informative, right? When it has been bombarded with many different things, maybe this is not a tissue to study. I am asked to do a profiling on very heavy radiation, and I'm saying, well, this time point, they are just going to collect dead cells. Don't bother the patients with the biopsy, so we need to brainstorm relevance of the tissue. Pathologists need to be on board, surgeons need to be on board, and industry need to step up because it's extremely expensive. And NIH is not, as we all know, NIH is not really also stepping up enough. They think that profiling has been done enough and we don't need to profile anymore. These are the type of critiques we have, in our NIH grant asking for profiling tissue. So it's a complex ecosystem. We need to change the culture. This technology is extraordinary. We need to bring it to the clinic and not leave it on the bench of, sophisticated, scientists. Need to bring it to the clinic.
Yeah, and I couldn't agree more. And Aviv, as you know, we've started these kinds of studies back in 2004 when I ran that first bladder trial with 10 patients and got so much pushback. How do you think industry can be brought to the forefront to say, hey, before you do an 800 patient trial, let's do a 10 or 20 patient small study that gives us information? How do we get the Genentechs of the world to, really fall behind us and support this concept?
We definitely do that. We're an early, we are a research and early development organization to begin with. And so our studies, our phase 1 and phase 2 studies, they are smaller, and our goal is really to understand, right? To know whether our drug is safe, of course, and then efficacious, but to understand it in high detail. And so this work for us is actually at the heart of what we do. Mm-hmm. But I will say more generally for Genentech and Roche is that we've been in the forefront of this kind of work. Going back to the days where there wasn't really single RNA sequencing, it was still mostly about genomic sequencing. We include consent for genomic research analysis in every trial. You often can't go back, even if you have the perfect trials with the perfect samples collected. If you have not consented the patients appropriately, tough luck now, there's no going back. And I actually have to be grateful to the people who came before me for having had the foresight to think about some of these things 5 years ago, 10 years ago, when it was less common to do trials like this, so that now we can come back to those samples and ask questions years later when the trial completes in the right way. And I strongly agree with Miriam's point about the importance of doing this across many different diseases. This is important for each disease on its own, but it's also, we have learned so well that disease mechanisms repeat themselves in very unexpected ways.
Absolutely.
CD20, very important to target in cancer, very important to target in MS. Two very different diseases, a big B cell component in both. That's a big lesson for a company like Genentech, of course, but it's a big lesson, I think, for us as a community, that you never know with biology exactly where the mechanisms would be. So everything that allows you to look across diseases strengthens you.
Especially the immune
other
system, right?
things.
Especially with the immune component that is going to be present across the same population.
Stromal cells and your epithelial cells, it's just true. And I mean, you're preaching to the choir with me on this and with my colleagues. We have learned, though, some important lessons. Samples are limiting. The better the technology is at getting you more in one assay, the better off everyone is, because there's so many different things that you can do. Which one would be the most impactful? In the end, the sample will be limiting. It's a biopsy. It's very small. There's only so much you can do with it. So that's where technological advances are important. And the clinical world is not the experimental lab in a research institute. Absolutely. So all sorts of things that you can arrange in a very convenient way, they fall away when it's a global clinical trial with dozens of sites, and you work by proxy through those sites until the samples are collected. And so you have to think very hard as a technologist, as a computational person, as a clinical expert, as a biological expert, to really make an operation like that successful. And that's where I think the partnership is so important, both in the early phases, as Pam highlighted, doing the small study right sets you up on the path to success. And in the late stage, when you have the very big trials, as Miriam referred to, doing those in a way that comes back and restarts the cycle of discovery so that we can find more, because, you know, there's still enormous amounts of unmet needs. And so that's for us a big commitment in Genentech. We have new departments for that, cross initiatives for that, lots of collaborations for that. And I hope others will do the same.
Absolutely. We need more people to follow Genentech. But let me just highlight the CD20 example that you give. So Zemin, maybe you can join this conversation, because I saw your data set about CXCL13, and I think that's very intriguing because, as you know, it's very much a chemokine that's going to attract B cells into that microenvironment to have that T-B cell collaboration, tertiary lymphoid structures. So do you think that maybe we need to start looking now at these cell neighborhoods in a more careful manner to dissect how each of these cell subsets and what's driving their trafficking into the microenvironment, whether it's hormonal, BMI, neuronal, all of these different signals that we have to start looking at?
Yeah, absolutely. I mean, so if you don't simply study the single cell without the neighborhood context, without the spatial information, maybe we are missing some of the key information. But now if you combine a single cell with the spatial data, it should actually become clear. I didn't have a chance to present some of our work here related to that, but TLS, the tertiary lymph structure is actually very important for the tumor in terms of regarding how they respond to different external therapeutic perturbation. And so those are, I think, going forward, if you look at CXCL13 is one of the manufacturers, but you can probably use different cytokines to somehow perturb or maybe enhance the TLS so that that location can be the place for generating more kind of pro-inflammation signals. So I think our affiliate company, Aviv, is actually taking some strategy toward this. I'm sure there are other places that are doing this as well. You know, I think, but I really want to, going back to the previous question, Aviv and Miriam just mentioned about the sort of having different pharmaceutical company involved in understanding human tissues. And we, you know, when I worked at Genentech for many years, but when I started my lab in Peking University, I found out actually it's very relatively easy from my end to get clinical samples, and we were able to form the kind of really tight relationship with companies like Amgen, Boehringer Ingelheim, and Bayer to really collectively study those clinical samples. We're able to get a large number of really high quality data, which are very beneficial for finding tumor-specific subtypes and therapeutic targets. And so those are kind of things that can happen in a pre-competitive space. And I imagine multiple companies like Genentech and Gilead, should probably participate in things like that, so that we can generate a huge amount of data that can be used. We have already shown that, in today's several talks, that such single cell and spatial data can be extremely powerful in identifying tumor relevant cell types and targets. And I think that this can happen definitely in a pre-competitive space, and some kind of correlation may be needed going forward.
Yeah, we're going to have larger data sets, so then we'll have to figure out how to get everyone to share their data sets, because that's the beauty of the strength in the computational algorithms, right? The larger the data sets, and then this bringing it all together from all the different groups, sharing it in a way that we can all use it. I think these are all pieces that we still have to put together. I'm going to switch.
The problem with industry, though, Aviv, I have to say, there is a need for a push to, putting the data out there. I have to say, even when you guys publish in journals, we are asked to put all our data out there, and industry is giving a pass. And I think that somehow this is not so useful.
I will just speak to Genentech. I can't speak for the industry as a whole, but Genentech publishes its data.
We need to
write our papers and we publish our data. And in that, we actually are often an outlier compared to a lot of others. But that's
we need that to be broad across.
I am a big believer in that as well, but I cannot speak to everyone.
Let me take a few questions from the audience now, if that's okay with everyone. Aviv, this is, I think, for you. Can we extend these new aspects of immunotherapy and systematic approaches towards liquid cancers, like lymphomas and blood cancers too?
So I did not speak to that, but it's not just can we, absolutely, and people have and are doing it actively. I'll highlight one additional technological features that can be applied in many liquid cancers and is harder to do in solid tumors. Well, first of all, they're often easier to monitor sequentially because of the systemic nature, and so easier to obtain samples and follow multiple times. But second, there's a lot of questions related to clonality in the cancer cells themselves in hematologic tumors. And their single cell analysis, which is a genetic analysis, has all sorts of extra tricks that you can apply. For example, we have shown that you could use the mitochondrial genome and transcriptome in order to identify clonal mutations that have nothing to do with the cancer itself, but helps you identify the clonal relationships inside the tumor and how they evolve under treatment, which is tremendously beneficial for understanding both the original tumor and mechanisms of resistance. So that is an example of work that has been done, and lots of these efforts were led by Vijay Sankaran from Children's in Boston. But there is a lot more, and these approaches fit very well, very, very well in that world.
Okay, well, I think maybe I'll try one last question, Miriam. If we give a short answer, we'll see. What is the duration of time needed to fully understand the mechanism of tumor formation and resistance for a given cancer? And I think it's an important question, because sometimes we're looking at a single time point, and it really is a duration of time that forms that. But maybe a brief answer before we finish up.
Well, that's a complicated question that we are all struggling with. I mean, we need to understand this dynamic of tumor, but it goes back to our modeling question that we were asking is, how do you best model? I think the way to approach it is what I think Zemin was also referring to, is really trying to understand, going across tumor, across different tissue type, and really trying to build, this commonality of cues, which he identified so well. And that will help us understand, some pattern that seems to be always present, as he showed, or whether, patterns that are specific to tissue type. And this is how we are going to build this big puzzle. These are complicated questions that, many of us are addressing experimentally in animal models, much more difficult to address in patients.
So Janine, I think I'm going to turn this back over to you at this point.
Yes.
I think it's been a really robust discussion. Thank you.
Absolutely. Thank you. I just want to say thanks so much to you, Pam and Aviv, Miriam, Zemin for a really fantastic session today. The talks and the discussion were really rich, and I'm sure everybody really loved it. Thanks as always to our audience for joining us from around the world. Please take a moment, if you have one, to complete the very short survey that I've linked in the chat box. We always look forward to your feedback. And don't forget to join our next Life Science Across the Globe event, which is taking place on April 6th at 9 a.m. Eastern Time, on the topic of new chemistry for biological systems. And that is being hosted by NCBS in Bangalore. So we hope you'll all come back to join us.